Buenos días, bienvenidos y bienvenidas a un nuevo seminario del ciclo de seminarios del Instituto. Hoy tenemos el placer de contar con Marta Martínez. Bueno, como habitualmente sucede, Marta dará su seminario en inglés, pero yo voy a hacer la presentación en español para no robarle mucho tiempo. Marta es científica titular desde el 2019 en el Instituto de Investigación en Ciencias de la Alimentación, que más a partir de aquí llamaremos CIAL, que es un instituto mixto del CSIC y la Universidad Autónoma de Madrid, que está en Canto Blanco. Pertenece al grupo de proteínas alimentarias. Marta, que es procedente de Valencia, doctora de Ciencias de los Alimentos por la Universidad Politécnica de Valencia, la UPV, y realizó su tesis doctoral que defendió en 2013 en el Instituto de Agroquímica y Tecnología de Alimentos, IATA, que también es del CSIC, localizado en Valencia, especializándose en el desarrollo de materiales biobasados con aplicación como envases alimentarios. Posteriormente, en 2013, comenzó un postdoc de 4 años en Australia, en la Australian Nuclear Science and Technology Organization de la Universidad de Queensland, donde se centró en el estudio en la estructura multiescala de paredes celulares vegetales utilizando técnicas avanzadas de scattering y difracción de neutrones y rayos X. En 2017, con un contrato, Juan de la Cierva volvió al centro IATA en Valencia, donde había realizado la tesis, y en 2019 obtuvo una plaza de científico titular en el CSIC, donde se incorporó en 2020 al CIAL, aquí en Canto Blanco. Con respecto a sus intereses científicos, se pueden resumir en que Marta y su grupo desarrollan estrategias encaminadas a una utilización más eficiente de los recursos naturales en alimentación, mediante un conocimiento exhaustivo en técnicas avanzadas de dispersión o scattering de rayos X y neutrones, posicionándose como una de las científicas más destacadas en España en la aplicación de estas técnicas en el ámbito de la ciencia de los alimentos. Ha realizado multitud de estancias en centros internacionales, varias veces en Suecia, en Alemania, en Estados Unidos, y tiene una producción muy prolífica con más de 90 artículos científicos en revistas indexadas y capítulos de libro. Es autora de 7 patentes y cofundadora de la startup Aerofibers Technologies. Prueba del carácter disruptivo de su investigación muy destacable es el proyecto ProDigest, el cual representa el primer proyecto ERC Consolidator en el área de ciencia y tecnología de alimentos concedido en el CSIC. Así que, Marta, gracias por venir.
Muchas gracias. ¿Se me oye bien? ¿Se me oye bien ahora? Sí. Okay. So I will give my talk in English in case someone does not understand Spanish. And first of all, I would like to thank you all for being here today. It's a pleasure for me to be here, and I'm sure we can find many common interests between the work that you are carrying out here in your institute and what we are doing at CIAL. And today I would like to tell you a little bit about how the small angle scattering techniques that we are using for our research can be useful within the food science area. And first of all, well, this we have already discussed. Javier has told you about what has been my trajectory in science. So I would only like to mention that why I started using the scattering techniques in the food science area. And that's because during my postdoctoral period, I was working in a nuclear reactor in Australia. And at that time is when I found out that these techniques, X-rays and neutrons, are extremely useful and powerful for investigating the structure of food systems. And so when I came back to Spain, I decided to continue fostering the application of these techniques in the food science area. And so before we start, I would like to highlight why food structure is something so important, something that we are interested in studying. And you can think of many common foods that actually present a very complex structure. We can think of natural substrates such as fruits, vegetables, cereals, and they have a very complex structure in their cell walls, as you can see here. We have cellulose combined with some other polysaccharides forming a very complex structure. But then we also have some products which contain a more or less complex structure deliberately. So to obtain products with some functional properties, texture, or organoleptic properties for the consumers. And then we can also produce different types of structures so that we can obtain, for instance, foods with texture. In the case of hydrogels, you can think of the gelatins or sauces, or some products we have some viscosity. We can also think of emulsions and emulsion gels, like to substitute animal fats in food products. And also for food packaging materials. Structure is very important, of course. And we can think of the conventional packaging structures, but also some more innovative structures, such as the aerogels, which are useful to develop these absorbent pads that we find in food packaging. So this is why we are interested in studying food structure. And now what happens in the case of food when we are trying to apply conventional techniques to investigate the structure? You can think of many microscopy techniques that are, of course, very useful to investigate food structure, but if we think of the native structure of many of these foods, we typically find biomacromolecules like carbohydrates in a highly hydrated state. And therefore, some of these techniques, some of these microscopy techniques require some sample preparation which is going to affect the structure of the sample. So we have to be very careful because sometimes what we are observing is not representative of the native food that we want to characterize. And in this sense, scattering techniques are very powerful because they allow us to investigate the structure of food systems, but also biological systems in their native state. We don't need to dry the samples, and we can also simulate relevant conditions for industrial processing and physiological processes, such as gastrointestinal digestion, for instance. So now, maybe some of you are familiar with scattering techniques, maybe some of you are not familiar at all, so I would like to briefly explain what are the advantages and the main principles of these techniques so that you can understand what I'm going to talk about. First of all, we can think of microscopy techniques, and here what we are doing when we are characterizing a sample is using lenses to generate an image of our sample. And we are typically focusing on very small areas of our samples. So sometimes it is possible that what we are observing is not completely representative of the whole or the bulk sample. In the case of scattering techniques, what we are doing is investigating the change in the direction of a source of radiation after interaction with our sample, and they are going to provide us some information on the bulk structure of the sample. So in this case, this is an advantage as compared to microscopy, for instance. And here you can see like a very simple scheme of what we are doing when we are conducting scattering experiments. We are using a source of radiation, which can be X-rays or neutrons. And what we are analyzing is what is happening to this radiation after it's interacting with the molecules in our sample. And this radiation is going to be scattered in different directions, so forming different angles. Then, depending on the position of our detector, we are going to register the intensity, the scattering intensity, and this scattering intensity, the angle that we are registering is related to the real sizes in our sample. So in this sense, if we use different configurations, if we move the detector to different positions from our sample, we will be able to investigate different size ranges. And you can see here that we have different techniques which are going to allow us to cover different size ranges. We have wide-angle scattering techniques, which are useful to investigate structures at the atomic level. For instance, I'm sure that many of you are familiar with these techniques to evaluate the crystalline structure of proteins, for instance. And then if we get closer to the sample, if our detector is closer to the sample, we are going to register smaller angles, and this is going to allow us to get information on larger structures. So this is going to be useful, for instance, to investigate how polymer chains are arranged in different types of systems. And we have also ultra-small angle scattering techniques, which are going to cover even larger sizes. And here you can see how we can combine these different techniques to cover different size ranges with interest in different applications. Apart from having the possibility of covering different size ranges, we also have the possibility of using different sources of radiation. And why is this important? Because X-rays and neutrons are not going to interact in the same way with the atoms in our sample. For instance, X-rays are going to be more sensitive to variations in crystallinity. So if we have crystalline and amorphous domains in our sample, X-rays are going to be very sensitive to this, whereas in the case of neutrons, we can play with the contrast that we will find when having hydrogen or deuterium atoms in our sample. This is very interesting in the case of foods, because as you know, in foods, we have a very large amount of hydrogen atoms, and what we can do is take advantage of this and substitute some of these hydrogen atoms with deuterium so that we can generate contrast between different components in our sample. And this is what is known as contrast variation. It's a methodology that is widely used in the neutron scattering experiments. And for food is very useful, for instance, if we want to see what is happening to the structure of polysaccharides and fats independently, for instance. So this is what I was talking about, generating contrast between different phases in our sample. So for instance, if we have proteins and polysaccharides or fats, you can see here that these molecules, they have different scattering intensity. This means they scatter the radiation differently. And as a result, they are going to provide different intensity. So we are going to register different intensity in our detector. And as you can see here, if we know what is the amount of substitution that we need to provide in our sample to mask the signal from the proteins or the lipids or the polysaccharides, we can even make some of these components invisible to neutrons, while the other one becomes more visible. And this is a very great advantage. So we can deliberately evaluate what is happening to one component in our sample when we are processing or we are doing any type of modification in the sample. And finally, what we have to do when we are thinking about conducting scattering experiments is not so simple, in the sense that it's not like other techniques. You prepare your sample, you run the experiment in the machine, and then the analysis is relatively easy. In this case, you have to plan in advance the experiments, because we don't typically have access to this type of instruments. We have the synchrotrons for X-rays, we have nuclear reactors and spallation sources for neutrons, and in some cases we have some lab equipments for X-rays, but it's not so easy to access these techniques. So we need to have some previous information on our sample so as to design very well our experiments. We need to apply for beam time. And then, if we are successful, we carry out our scattering experiments, And then we need to process our data. This is more or less complex. We need to have some background on these techniques to be able to extract the structural information. And then we can also perform modelization for, I want to know what type of structure this protein is presenting, so I can apply globular models, or I can apply like lamellar structures, or whatever you can think of. You can find theoretical models that you can try to fit to your experimental data. So that would be the process when we are conducting scattering experiments, and as we were discussing, there are several advantages. These techniques are non-destructive, except from the synchrotron experiments. In some cases, we have to be very careful because we can produce radiation damage in our sample. We can obtain information on the bulk properties of the sample, and we don't need any specific sample preparation. We can run samples, biological samples, food samples in their native state. And we can also generate contrast between different phases in the samples. So why are not they being so widely used in the food science area? Because we have several disadvantages. They are not perfect, of course. So they are very, they are expensive techniques. So we typically need to apply for beam time in these specific facilities. They are not so accessible. For some of these techniques, the size of the sample can be too large, especially when we are working with biological systems. That can be a problem. And of course, the data processing and interpretation can be somehow complex if you don't have the required background. So I think the idea is to combine these techniques with other complementary methods that can provide and support all the information that we are obtaining with these experiments. That would be the approach, I think. And here you can see as an example how we study the very complex structure of plant cell walls by combining scattering techniques, wide angle scattering techniques, and small angle and ultra small angle scattering techniques with microscopy and spectroscopy to find out how cellulose was organized in a hierarchical structure. So starting on how cellulose molecules are forming semicrystalline domains, and then they are arranging into individual nano or microfibrils and combined with other polysaccharides to form the plant cell wall structure. And so, what we have here in Spain available is the Spanish synchrotron. We have stacked wax beamline, so that we can perform simultaneously both techniques. But we can also find other facilities in Europe that have access to X-rays and neutron scattering techniques. So you can apply for beam time in many facilities. That is not a problem. And as we were discussing, they are not popular in the food science area. And mainly that's because you need to have some background more related to physics, which sometimes is complicated. However, they are very powerful and they present very important advantages. I think the possibility of analyzing the samples in their native state and also simulating processes or physiological conditions is something very important in this area. So now let's see some examples applied to the food science area. We have a problem right now because, as all of you know, prices for foods are rising and rising. Sometimes it's very complex to buy nutritious foods at an affordable price. And this is going to get worse with time, because we know that natural resources are scarce, and we are consuming more and more. So in this context, the food industry and scientists, we are trying to find alternative sources of food so that we will be able in the future to provide sustainable and nutritious foods to everyone. And so these novel food sources need to be nutritious, they need to be accessible to everyone, and they need to be sustainable. And what we are observing in the case of proteins, for instance, is that the trends are towards replacing at least partially the animal-based proteins with plant proteins, but also with alternative proteins, insects, seaweeds. We are also hearing a lot about cultivated meat. And in my case, in my group, we are working a lot with seaweeds, because my background on plant cell walls and plant biomass made me think that this could be a very interesting substrate for novel foods. But now, what is going to happen with all these novel foods? Do we know if they are safe for human consumption? What are going to be the implications in terms of nutritional quality and health impact in the consumers? We don't know. So at this point, it is clear that we need a methodology to correlate food structure with digestibility and bioavailability. So how are we going to digest these foods, and what is going to be the health impact of these foods in the consumers? And we know that when we eat the foods that we are intaking, they are going to be processed in our organism. They are going to be hydrolyzed and digested by some enzymes in the gastrointestinal tract. So we are going to generate digestion products, some molecules that at the same time are going to be transported through the intestinal epithelium, and they are going to be producing some metabolic responses. And we have in vitro methods to study this part of the digestion, to see what is happening, how the foods are being hydrolyzed. And we also have in vitro cellular models to investigate the intestinal transport. But then we don't know what is happening in between those two processes, because all these digestion products, how are they interacting with each other? How are they interacting with physiological components? And what type of structures they are forming so that they can be transported in the intestinal epithelium? We don't know. So in the case of proteins, we know that during digestion they are hydrolyzed and they form peptides and amino acids. And what we don't know is, I was commenting, we don't know how they interact with each other, these peptides or amino acids, and how they interact with components in the physiological medium, such as, for instance, the bile salts. But the hypothesis is that this nanostructural assembly, so how they are combined and the type of structures that they are forming, is going to be highly relevant to how these nutrients will be transported. And so we started this last year an ERC project in which we are going to study how the digestion products from food proteins are interacting with each other, what type of structures they are forming as a result of the interaction with physiological components, but also with other components that we may find in food substrates, for instance, fibers, which are highly abundant in these alternative sources, such as seaweeds. And then the idea is that we start with simplified systems in which we have these food proteins combined with fibers. We see what is happening in terms of nanostructural assembly, but then we move towards more complex systems, whole foods. And we also compare these in vitro digestions with in vivo digestions. Of course, in this case, a very limited number of studies, because what we want to do is to reduce as much as possible the use of in vivo models. And finally, correlate all this nanostructural assembly with the bioavailability and bioaccessibility of the nutrients, so that we can finally predict and modulate the digestibility and bioavailability of novel food sources. And the traditional approach to study the digestion process has been to study structural changes at the molecular level. You can think of peptidomics, which are very useful to understand what is happening to the proteins during digestion, what type of peptides are being generated during this process. But what we want to do now is to combine these techniques, which are going to provide information at the molecular level, with scattering techniques to cover also the nanostructural range. So we want to know how these peptides are forming possibly micellar or lamellar structures, and also larger structures, like in the case of gels or emulsions. And for that, we are conducting experiments at the synchrotron, and also we are applying for beam time in neutron facilities across Europe, but also all around the world. As I told you, I was doing my postdoc in Australia, so probably we will go there also to perform neutron scattering experiments. And now let's see the particular case of seaweeds. I was telling you that in my group we are working a lot with seaweeds, and that's because we think they are very interesting as a novel source of proteins. And why is that? Well, first of all, if we compare seaweeds with plant proteins, they do not require the use of fertilizers or water. There are some species that can be cultivated, even though some others are harvested naturally from their habitat. Some others can be cultivated, and they have very high growing rates. They are rich in fibers, they are a good source of proteins, but they can also contain other interesting compounds, such as vitamins and minerals. And of course, they are traditionally consumed in Asia as a source of food. It's just in Europe that we are not really used to consuming seaweeds as foods. But they also have some drawbacks that make them still not fully exploited in the food industry, at least here in Europe. And it's because the proteins are highly unexplored. We don't know anything about the structure of the proteins that we find in seaweeds. We don't know how they are digested. And also, one important thing is that there are no regulations in Europe with regards to the maximum amount of some components which could be, in some cases, problematic, such as iodine. So there are some recommendations in some countries, such as France, but there are still no regulations in Europe. So let's start with the fibers. How we can valorize the seaweeds to use their fibers and to formulate new food products. In seaweeds, we find several polysaccharides, which are sulfated polysaccharides, and they have the capacity of forming gel structures. So these gel structures contain the polymeric chains, and they also contain a large amount of water in their structure. You can think of the conventional gelatin, for instance. So that would be something similar. But we can also substitute part of this water with a lipidic phase, an oil, for example, and then we can produce a different type of structures which are known as oleogels or emulsion gels. And in my work, in my group, we worked on the development of these type of structures to substitute animal-based fat in food products. But they can also be used to other applications, such as biomedicine, or to produce controlled release systems in which we have lipophilic bioactives. And as you can see, we can control the morphological properties and also the functional properties of these structures by affecting different or by changing different components. We can modify the gelling matrix. For instance, we can use different polysaccharides or sulfated polysaccharides in this case. We can create different types of interactions between this gelling matrix and the components in the whole system. And we can, of course, modify the content of the lipophilic phase and also modify the type of oil that we are incorporating. And all of this is going to affect to the final structure and the final functional properties. And why we wanted to substitute the animal fats in food products, especially, you know that the saturated fats, they have been proven to have not very good effects on health when they are consumed in large amounts. So we wanted to at least partially reduce the amount of these hydrogenated or saturated fats in some products using these emulsion gels. And we developed several prototypes of meat products in which we substituted partially or completely the animal fat with these emulsion gels. And we had very promising results because the properties, not only the texture, but also the organoleptic properties in general were very good and acceptable according to the consumers. But now, let's go back to, okay, we have developed these new products, but how are they going to be digested and how are they going to be processed in our organism? What is going to be the health impact of these novel foods? And in this case, we wanted to study what would be the effect of incorporating, for instance, bioactive compounds in the lipophilic phase, the presence of these different gelling polysaccharides, and the type of gelling network that we were developing, what was going to be the effect of all of this on the digestion process. And to do that, what we did in this case was to develop like a control system in which we included nanoemulsions. We have no gelling matrix in that case, only the oil phase, which is dispersed. And then the emulsion gels, and also another alternative in which we remove all the water from the system. We only have the oil dispersed in the polysaccharide phase. And these type of systems, they are called oil-filled aerogels. So there is no water at all. And then we subjected these different types of systems to the gastrointestinal digestion process using an in vitro method, simulating all the physiological conditions with enzymes, salts, pH conditions. And we studied what was happening in terms of nanostructure using small angle X-ray scattering, in this case, SACs. Okay, so first of all, as we were discussing previously, in this case, we used two different types of polysaccharides. You can see here on the top, these are the emulsion gels which contained carrageenan as the gelling polysaccharide. And these ones are exactly the same formulation, but then the polysaccharide is agar instead of carrageenan. And you can see that only by changing the polysaccharide, the structure is changing completely. We have a different distribution of the oil droplets, but also the texture and rheological properties are completely different. And then if we remove the water phase, this is from the carrageenan, so these ones, we remove the water, we have also very different type of structures and they behave completely different. So now let's see what happens when we digest these different structures. When we digest these samples, because the polysaccharides are non-digestible, so we don't have, in our organism, we don't have enzymes which are able to break down these polysaccharides. So they are going to remain more or less intact after the whole digestion process. And therefore, what we find when we finish our whole digestion process is a solid phase, which is rich in these non-digestible polysaccharides, and a liquid phase, which contains all the digested material. And so we analyzed separately what was happening to the solid phase and to the liquid phase. In the solid phase, as we were expecting, we mostly have the polysaccharides, and in most cases, the structure of the polysaccharide was not being affected at all as compared to the initial sample. So, as we were expecting, enzymes, pH is not doing much to the polysaccharides. Only in one case, we saw something interesting, but it's not related to changes in the polysaccharide. It's related to part of the oil being retained within this solid structure. When we were using carrageenan as the gelling polysaccharide, part of the oil was not being digested, and it was held within the solid structure. But then, if we look at the liquid phases, so that means what has been digested, we find something very interesting, and we find different structural features that correspond to different types of nanostructures that are being formed as a result of the digestion. So first of all, we observe one scattering feature which is related to the presence of bile salt lamellae. This structure we can find when we have suspension of bile salts, but then these structures are being affected by the fatty acids that are being produced during digestion. So we find out that depending on the starting material, the amount and structure of this bile salt lamellae is changing significantly. And also depending on the degree of digestion, so if we have more or higher amount of free fatty acids being released, such is the case of nanoemulsions, we have a higher formation of these type of structures. But then we also see another feature which corresponds to larger structures. And in this case, we believe that this corresponds to less digested oil phase, like triglycerides, which are being combined to form this type of vesicle structures. And we cannot find these structures in all the cases. We see that these structures are more abundant in those samples in which we incorporated a bioactive, which is curcumin, in this case into the oil. And so it is very important because we only incorporated curcumin in a very, very, very small amount into the oil, but this was having a very large impact on the digestion process, which is very interesting. And so you can see here the different types of mechanisms that we proposed for the digestion of these samples. Depending on the type of polysaccharide, we have more or less formation of these vesicular structures when we have an oil phase which is less digested, or if we have a high degree of digestion, then we will find mostly these lamellar structures. So this is interesting because that means that if we want to have a more digestible food, we can choose this type of polysaccharide, carrageenan in this case, or if we want to have less digestion, we can choose agar to develop these structures. And then another example, which is more related to what we are doing now, we are also valorizing these fibers to develop structures in which we want to modulate the digestion of proteins. So for instance, we want to increase or reduce the degree of digestion of a protein such as casein that we know during the gastric phase, so in the stomach, is highly digested, but we want to make it able to reach the intestine. What can we do with this? So we can incorporate this casein into hydrogels, and so we have a protective matrix of the polysaccharide, which is going to affect. And so this is just to prove that at the end of the gastric digestion, we have some protein which is not digested, but then most of it is digested after the intestinal phase. but we are able to produce the release of peptides with larger size. And this can be interesting, for example, if we want to produce foods with a higher satiating effect. And again, what is happening during digestion? So what we did was exactly the same approach. And we compared what was happening when we have the casein alone or when we have the casein in the presence of fibers, just in blends, like we blend the casein with the polysaccharide, or we incorporate the casein into hydrogel structures. We perform the same in vitro digestion protocol, and then we analyze what is happening on the solid and the liquid phases. And this is the typical scattering patterns that we get when we analyze the structure of casein, so that you can see that a very well-known protein, such as casein, that you can find in milk products, it contains a very complex structure. So before we digest the casein, it is organized into different structural levels. We have the calcium phosphate nanoclusters, then these are combined to form submicelles, and we also find micelles. So we have different structural levels in the protein. And when we digest this protein, you can see that we also find different structural features. So what is happening? During the gastric phase, what is happening to the protein is that the enzymes are penetrating the micellar structure and they are swelling. So they are disrupting the structure of the protein from the inside. And then at the end of the intestinal phase, we find similar structures as we observed in the case of the emulsion gels, if you remember. So we have this bile salt lamella, and these structures are also combined with vesicular structures. So something similar is happening during digestion. And I'm not going into detail, but this is that all the information we gathered from the small angle X-ray scattering experiments allowed us to propose a mechanism for the digestion in the different types of systems that we were analyzing. And this is providing us information on how to design these products so that we get the degree of digestion that we want to induce and to release, for instance, larger peptides to make the food more satiating. And then finally, something very interesting. So we are talking all the time about the bile salts and how they can form these lamellar structures and interact with the digestion products. So the next thing we wanted to see was what is happening if we are digesting the protein at different concentrations of bile salts. How is this going to affect the digestion of the protein so that we can understand what is happening at the nanostructural level? So first of all, you can see that if we digest the protein alone, bile salts are inducing a higher degree of hydrolysis of the protein so that we have higher amount of free amino acids. You can see also here by TEM that the type of structures that are being formed are completely different, and that's because bile salts, they also have the capacity to act as biosurfactants. So this is going also to affect to the formation of larger structures. And finally, if we look at the nanostructures that are being formed by SACs, what we see is in the case of casein, it is so highly digested that we are going to find the same type of nanostructures regardless of the concentration of bile salts. But that's because the protein is already very digested. But what is happening when we are digesting the protein in the presence of the fibers? We saw previously that the fibers are able to delay or reduce the degree of hydrolysis of the protein. And how is this going to affect to the digestion process? So first of all, we checked what was happening when the protein was incorporated into hydrogels. And as you can see, this is the solid phase, so the non-digested phase after the whole process. We can still find some protein which is retained in this solution, in the digested fraction. But this amount is decreasing as we increase the concentration of the bile salts. So the bile salts are inducing the release of the protein towards the digestion medium. As we observed with the casing, the same is happening in this case. The bile salts are also affecting to the type of structures that are being formed at a larger scale. And what we can see is that we have less amount of digestion products, but larger structures when we increase the amount of bile salts. And very interestingly, when we look at the profiles, what we see is that only for the larger concentration of bile salts, we have an increased amount of these lamellar structures. Our hypothesis is that what is happening is that at this concentration, bile salts are increasing the diffusion of the protein towards the liquid medium. This protein is being more digested, and this protein, together with some polysaccharide which is also being released, they are stabilizing these lamellar structures. And finally, when we digest the protein in the presence of the polysaccharide, but with no particular structure, so we only have physical blends. If we look at the solid phase, we don't have so much protein which is non-digested as it was happening with the hydrogels. We also see that the type of structures which are being formed at a larger scale are completely different, because in this case we have the polysaccharide solubilized. And interestingly, because we have the polysaccharide solubilized into the medium, what we see is that there is a much larger proportion of these lamellar structures which are being formed. So that means it is very relevant that we have an amount of soluble polysaccharides during the digestion so that they can stabilize these lamellar structures. Now, we don't know how this will impact on the transport mechanism, but for sure it will have some implications, and that's what we will study next. And finally, just as an example, I will show you also how the scattering experiments can be useful to study the structure of proteins from the seaweeds during digestion. We have been working with different types of seaweeds with different species, and the composition is completely different, so the nutritional properties are going to be also very different, and the protein content is significantly different. So I will show you some work that we have done with these seaweeds using this in vitro digestion protocol. And what we did in this case is very interesting. We performed these digestion experiments at real time while we were measuring the samples on the synchrotron beamline. So as we were conducting the in vitro digestion, we were continuously collecting SACs and WACs data from our sample. And what we can see, we still have to analyze this data in more detail. What we can see is that in the case of this seaweed, which is more digestible than other species, as you can see, because the cell walls are more labile, what we can see is during the intestinal digestion, we find these lamellar structures that are being formed, and they are changing with time. So we now would like to see what is happening during this whole digestion process, but I think it's very interesting for you to see that you can simulate, this is digestion, but you can simulate any food processing in the beamline and collect this data to see what is happening at real time. And so to conclude, I hope today you could see some examples of how these techniques are useful for the food science area, and maybe you can think of applications in your areas. But I would say that small angle scattering techniques are very powerful to investigate the native structure of foods and also the nanostructure assembly of foods during digestion. The digestion products, we have seen that they are able to interact with each other and with other components from the physiological medium, and they are forming different types of nanostructures. And this, we expect that will have a significant impact on intestinal transport and ultimately on the health impact of these novel foods. So hopefully all of this knowledge will be very useful to develop new food products, but also to some other applications such as the development of bioactive compounds for pharmaceutics, also to see how we can relate the structure of different proteins, different novel proteins and their digestion mechanism with their allergenicity, and also for several biotechnological applications. So I hope this was interesting to you. And with this, I would like to thank you all and all the people from my group and the scientists from the different facilities that helped us conducting these experiments. Thank you.
Thank you, Marta, for your beautiful talk. Now we may have some time for questions or comments.
Thank you, Marta, for the nice, both, I would say, scientific and engineering approach that they sum up. And I was wondering, when you refer to oil, what do you mean with oil?
Oil.
What type of oil?
We have been working with different types of oil, but some examples: sunflower oil, rapeseed oil, coconut oil.
So they are characterized in composition and so on?
Yes.
Okay. And then I was just wondering whether you are able, it was kind of a dream many years ago when you were using SACs for studying the lipid polymorphism. And you were having the micelle, the bilayer, H2 phases, and cubic phases. Are those the so-called bilayers that you detect related to possible cubic phases? I mean, cubic phases, I don't know how you call it in one of the slides, are a kind of weird arrangement of bilayers that they look like an anemone or something like that.
So you mean the lamellar structures that we see in the digestion?
You had another one that contains, if you go to the one, I don't know how you call it, usually they were referred to, at the very beginning of the talk, they were referred to as cubic phases.
I have to say that I'm not an expert on fats, so this is, I mean, I have been working mostly with proteins and polysaccharides. Is this one?
No, at the very, 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 very beginning.
Ah.
When you were referring kind of to the lipid polymorphism that you could detect, you refer to it as a secondary whatever. Because it's not the typical flat bilayer. It has a 3D arrange.
I don't know which slide you. I know.
The typical flat, okay, the flat that you entangle like encapsulating structure. It's just kind of an octopus arrange of bilayers. That they are so-called cubic phases, and they have other properties which are far away from that of a bilayer.
I have no idea about these specifics. I guess for these type of structures, you would have to go to wide angle scattering because we are talking about crystalline arrangement of the lipids. And in that case, I guess it's a matter of finding models to try to feed your data and see what type of model is working best to describe the structure that you are observing in your sample. But like I said, I'm no expert on, lipid structure, so I cannot tell more than that. That you would have to find a fitting model and see if that helps in finding out the specifics.
Okay, and there is another question, and it's regarding how do you break the cell wall of a seaweed?
How do you break it?
How do you break it?
That's a good question. And that's what we are trying to do with some species which are they have a very recalcitrant cell wall structure. Some others, like nori, this was nori, you digest the seaweed, and most of the cell walls, they are permeabilized or at least partially broken down.
So they just explode by osmosis.
I don't think they explode. It's like enzymes are slowly digesting the components in the cell wall, and they are, what we have observed in some cases is that they are swelling. So it's like something similar as we observed in the casing, like the structure is being digested from the inside to the outside. It's like enzymes are, breaking down components in the interior part of the cell walls, and they are swelling and eventually breaking down. For others, not even with digestion, we see that the cell wall is almost completely intact at the end of the digestion. So in those cases, what we are studying now is how other processes like cooking, for instance, boiling, high temperature, or mechanical processing can help disrupting the cell walls during digestion. But it's true that it's a limitation for some species because they have very, very tough cell walls. So, yeah, of course.
Thank you.
Thank you, Marta. Really very nice talk and very interesting approach. I wonder, when you are mimicking the digestion of, human digestion, I understand. So, so in this case, what kind of enzymes are you using in order to simulate this? Is just the casein and these ones? Are you not using lipases for digestion of fats, or?
It depends on the substrate that we are digesting. In the case of the emulsion gels that contain oil, we are using lipases. But then when we are working with proteins and we don't have any fat, we are just working with pepsin and pancreatin, amylase if we have some polysaccharide which could be digested by these enzymes. But we have to be very careful because the enzymes are also protein material. They are going to affect to all the calculations that we want to do to calculate digestibility. They are also, they may be a problem for the scattering experiments because if we have a high amount of enzymes, they are going to interfere in the scattering signal. So that's why we only select the enzymes that we need for each specific substrate. And I know for whole foods, we will probably need all of them because we typically have fats, carbohydrates, and proteins. But since we are now working with these model systems or simplified systems, we are just using specific enzymes.
Yeah, because there is a really very nice connection with your kind of study. So we have seen many years ago that indeed the lipases are, the pancreatic lipases are making a complex with a cofactor, a protein cofactor called colipase. And then there is a micellar activation. So at the moment you have a micelle that is really small, empty of other components, fatty acids and so on. So this micelle is able to activate the complex, the lipase-colipase complex, and then all the products of the digestion of these fats are incorporated into these micelles in order to increase the size. At a specific size, this micelle is not longer being able to activate the complex, and there is an inactivation of the enzyme. So these micelles are acting as carriers in order to transport the fatty acids to the walls of intestine. So in this sense, I think you could use this system in order to see by other techniques how it is produced, because there is a regulation system in humans for fatty acid degradation.
Yeah, for sure. And it would be very interesting to perform the in situ digestions, because then you can see what is happening at real time. So if at some point these enzymes are inactivated and this complex formation is no longer taking place, you can study that by performing in situ digestions. So I think that would be very interesting.
Thank you.
No, any more questions? Yeah?
Hi, Marta. Thank you very much for the nice talk. I have just a little question. Is why you don't use, I just want to know why you don't use SACS instruments in laboratory scale?
First of all, because there is a very limited number of lab SACS instruments, and they are mostly, from what I know, no institute in the food science area has any of these SACS equipments here in Spain. Of course, overseas there are some research institutes that have access. One of the problems of the lab SACS equipments is that they require longer times than the ones you use in the synchrotron. For instance, you can measure one sample in the synchrotron in 10 seconds, and then in the lab SACS equipment you will need 3 hours for the same sample. That's because the intensity of the X-ray beam is not the same, of course, so that could be a limitation. But it would be, it's very useful, of course, if we would have access to these instruments. So
thanks.
Okay. There's no other question, then I think that we can call it a day. Let's thank Marta for her nice talk. And we would also like to give you this present from the Institute,
Thank you.
as appreciation for your visit. Thank you.
Thank you very much.